Hi, my name is Nate Viren. I'm a PhD candidate in Bruce Donald's lab at Duke University. I'm presenting today on an algorithm we've developed called Resistor, which is an algorithm to predict resistance mutations. I was planning on being there in person, and I wish I could be there, but unfortunately, COVID got in the way. So let me tell you why drug resistance is important. Two cancers that we spent a lot of time looking at over the past year in our paper are lung adenocarcinoma and melanoma. Over the past 2 decades, a number of drugs have been developed to target the epidermal growth factor receptor in lung adenocarcinoma and BRAF in melanoma. Unfortunately, because these are target-specific drugs, resistance mutations often occur, rendering the originally efficacious drugs obsolete in oftentimes less than a year. Our hypothesis is that if we had a better predictor that could accurately predict how resistance mutations would occur, we might be able to develop more durable drugs. To be able to design a predictor, we first have to understand that there's lots of different ways resistance can occur. There's point mutations, deletions, changes to drug influx, efflux, and other ways. In Resistor, we really concentrated on that first factor, namely point mutations, and we did so along 4 axes. The first is we looked at negative design, where we tried to identify mutations that ablated binding to a drug. We also looked at positive design, which is sort of the inverse, namely mutations that caused a protein to increase binding to its endogenous ligand. We also tried to identify mutations that would actually occur in different types of cancers, that's called mutational probability, and locations at which there are lots of these types of mutations, hotspot identification. In other words, we combined structural and genomic predictions to come up with a single resistor prediction. But what is this K-star positive and negative design that I mentioned in the previous slide? Well, to get there, I need to first tell you what OSPREY is. OSPREY is a software suite that we design in our lab at Duke. It stands for Open Source Protein Redesign for You. In OSPREY, we design and implement a number of algorithms for predicting biophysical properties about proteins and their interactions with other proteins and small molecules upon mutation. One of the main algorithms in OSPREY is called K-star. K-star is a provable approximation to Ka, or binding affinity. K-star works by computing partition functions over molecular ensembles of a protein-ligand complex in 3 different states, namely the complex state, the protein state, the apoprotein state, and the apoligand state. It's able to compute these approximations using A-star conformational search, and this A-star search enumerates these molecular conformations in order of low energy. By choosing this particular ordering, K-star doesn't actually have to look at the entire molecular ensemble. In practice, it actually only looks at a very, very small fraction of the total molecular ensemble while still preserving provability in the approximation. Resistor uses K-star to basically pose the following question. Can we use K-star to identify mutant sequences that cause an enzyme to increase binding to its endogenous ligand, which is positive design, or ablate binding to a drug, which is negative design? To get at both of these factors, because both are really important to resistance, we look at the ratio of the positive to negative design score. Whenever we see a ratio of a sequence increase compared to the wild type sequence, this is Resistor telling us that we're predicting this sequence to be a resistance mutation in vitro. Another criteria we use in Resistor is the probability that a mutation will occur in vivo. We call this the mutational probability. A lot of this work was done by our collaborator and paper author, Dr. Teresa Cazerer. So what are mutational probabilities? Mutational probabilities are derived from mutational signatures. Mutational signatures themselves are deconvolved from tens of thousands of whole exome and genome sequences across many different types of cancer. And the hypothesis is that a particular mutational signature is indicative of an underlying mutational process. So for example, lung adenocarcinoma here has 5 different mutational signatures in it. The largest mutational signature is signature 4 here, which has been associated with smoking. And we can see that signature 4 has a lot of C to A mutations. Another signature in lung adenocarcinoma is this pink one, which is signature 2, and it's been associated with ApoBec activity. And this signature has a lot of C to G and C to T mutations. We use this data to be able to ask questions like, what's the probability that threonine 790 will mutate to methionine in lung adenocarcinoma? Well, that's equivalent to asking the question, what's the probability that this base C will mutate to base T in this particular sequence context? To get at that, we compute the probability of that mutation occurring in each of the different signatures, add them together, and then come up with a single number that is the mutational probability prediction. We need to come up with a way to combine the different criteria that we just looked at, and we did so using Pareto optimization. So first, a definition. A protein sequence X dominates another sequence Y when every objective function when calculated on X is at least as great as that calculated on Y, and for one of them, it's greater. We say that a sequence is Pareto optimal when it's not dominated. So let's look at an example. Here we have positive design, or mutations that increase binding between a protein and its endogenous ligand. And here we have negative design, or mutations that ablate binding between a protein and its drug. We see that sequence E dominates sequence N because it is greater on the positive design axis, and sequence C dominates sequence K because it's greater on the negative design axis. But neither C nor E dominate each other, and they're both on the Pareto frontier. So in Resistor, we used 4 axes, namely positive design, negative design, mutational probability, and hotspot cardinality to compute a complete Pareto rank. So at this point, it might be helpful to just summarize exactly what the Resistor algorithm is. So from the start, we start with a positive and negative design upon which we're going to run K-star. Before we can do that, we need to get our structures and specify what sequences we're interested in, as well as what type of flexibility we want to have. If we were doing a combinatorial sequence search, there's a couple of algorithms in OSPREY that are really helpful for sublinear approximation and pruning, in particular COMETS and BWM star. In our RECOMB paper, we only looked at single point mutants, so we didn't have to use these algorithms. We then run K-star approximation on the positive and negative designs, and we prune those sequences for which the change in the K-star score ratio does not increase. We also prune those that our mutational probability calculation finds have no chance of occurring in vivo. So how do we calculate that aspect of it? Well, we start with the coding DNA for the protein of interest, and then we scan over the sequence looking at every codon along with its 5' and 3' flanking bases. And we assign those probabilities that we calculated earlier to each potential mutation in a particular cancer type. And to do that, we used a new graph algorithm that we wrote, where the vertices of the graph are the codons of interest, and then the edges are the mutational probabilities between each of those codons. From that, we're able to calculate the probability that, for example, alanine 692 mutates to valine in lung adenocarcinoma, and all of the other potential mutations. And we combine those predictions along with the K-star predictions and the hotspot cardinality predictions using Pareto optimization to rank every potential resistance-causing sequence. So let me give you an example of one that we actually did. We looked at 3 drugs that have been developed for treating lung adenocarcinoma that target EGFR. And what we found after running our Resistor on these 3 drugs is that Resistor correctly predicted 8 resistance mutants for these 3 drugs. And excitingly, 7 of those 8 resistance mutations were on the Pareto frontier. One of them, L792H, was actually ranked on the second Pareto rank. Let's take a closer look at that particular example. Here on the left, we see in this dark blue here, this is the wild-type leucine, and then in the magenta here, we have the histidine mutation. And what we can see by looking at the middle is here with the space-filling representation is that histidine mutation really crowds osimertinib. which forces osimertinib here in sort of this dark pink to translate out of the binding pocket and probably lose favorable interactions with EGFR. But we're also really excited about the potential of using Resistor in a prospective manner during the drug development process. And so we wanted to come up with a couple of predictions that we could actually test. To do that, we collaborated with Dr. Eduard Stefan and Andreas Feischner at the University of Innsbruck, and they've developed a biosensor called KinCon, which stands for a kinase conformational reporter assay, and it's a protein fragment complementation assay that looks at full-length kinase domains and attaches a half of a luciferase on the N term and half of a luciferase on the C term. And this biosensor works because these kinases have an active conformation, which is an open conformation, and an inactive conformation, which is a closed conformation. When the conformation is active or open, they measure very little light, but when it's inactive or closed, they measure a lot of light. So that measurement of light is a proxy for the enzymatic activity of these kinases. We came up with a set of predictions we are interested in interrogating for two reasons. We looked at first those that had differential resistance predictions across different drugs. Here, the hyphen means that we predict a particular drug will remain effective against on a particular mutation, and if there's a number, that means that's the Pareto rank that we predict resistance to occur for that drug. And more importantly, we also chose mutations that were present in patient populations. So we looked at the COSMIC and C-BioPortal databases, and in the last two columns are the numbers of patient samples that actually had these particular mutations of interest. Up on the top right here, what we're looking at is the control for the change in the relative light unit, LRU, for each of these different mutations of interest. Everything is normalized to the wild type BRAF, and what we see here is that this V600E mutation, which is sort of the canonical BRAF oncogenic mutation, the LRU drops to about 0.4-fold, meaning that it adopts a much more active conformation. Likewise, for G466E and G466R, we have a similar opening and activation of the kinase. For these other three mutants, we didn't see much opening of the kinase, and so that's relative because that's important when we consider that we made predictions around assuming these kinases would adopt an active conformation. And you'll see why in just a minute. What we found is that Resistor was able to correctly predict 75% of the particular sequence and drug combination effectiveness for G466E and G466R. So on the left here, this is again the V600E control versus the wild type, and everything is normalized to the untreated DMSO molecule. And what we see here is that on the wild type, there really isn't much closing when drugs are added to the wild type kinase. But when drugs interact with the V600E kinase, there is much more closing, which is why these drugs are inactivating for the V600E mutation, which is what you want to see. So excitingly, for G466E and G466R, we found that all four drugs continued to work for those two mutations. So we got 3 of the 4 drugs correct. The one we didn't get right was dabrafenib, and we think we didn't get dabrafenib right because we probably started with a structure that wasn't exactly correct. And when we do these structure-based molecular predictions, your starting structure is really critical, and everything is sensitive to the starting structure. We didn't see much effectiveness for the drugs for these other 3 mutants, and that sort of makes sense because these drugs target the active conformation, and we just saw on the previous slide that those mutants aren't activating mutations. In conclusion, we've designed a new algorithm for predicting resistance mutations. Resistor uses multi-objective optimization over structural and sequence data. We've used Resistor to predict new resistance mutations that we've experimentally validated. We've also predicted structures for these mutations. In the future, we'd like to apply Resistor to additional cancer types and drugs, to the antibiotic, antifungal, and antiviral spaces, add additional resistance-causing criteria to the optimization, and we're always looking for algorithmic improvements. I'd like now to thank my fellow authors on the paper, Dr. Bruce Donald at Duke, Dr. Teresa Kazerer, Dr. Eduard Stefan, and Andreas Feischner at the University of Innsbruck, and of course, the generous support of the funding agencies, the NIH, and the Austrian Science Fund. OSPREY and Resistor are free and open source software. You can download it at the link at the bottom right of your screen. So try it out and let us know what you find. With that, I'm now ready to answer any and all of your questions.